This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 722, recorded on February 19, 2021. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dixon Despommier.
Howdy, Vincent. Well, looking out the window, it's schniggling. The snow is coming down as though it were sleet and hail and anything else but snow. It's hard to see it, but if you stick your hand out the window, you can feel it. So it's been a dreary couple of days, don't you think?
Yeah, it snowed yesterday. It snowed today, but at least it's cleared up. Before, I couldn't even see the river.
Yeah,
Dixon.
that's right. That's right. It was foggy as well.
Also joining us from Austin, Texas, Rich Condit.
Hi, everybody.
Speaking of weather.
36 degrees. It's sunny right now. It's thawing a little bit. We've had, as many people know, a solid week of nighttime sub-zero temperatures that has caused a lot of damage. So
you got snow on the ground?
Yeah. It's
so you said you don't have any water, but you could go and melt some and at least feed your plants.
Trust me, the kitchen is full of buckets and stuff.
Okay.
Our primary concern is keeping the toilets running.
Right.
And
that's right.
So that, I mean, we got plenty of stuff around to drink. We got plenty of food. We were well stocked. I'm glad to run the pantry down for.
You buy another couple of hair dryers.
Tell you the truth, but there are a lot of people that are much worse off than we are. So I'm grateful for what we have.
Well, I'm glad you have electricity so you can do TWiV today.
Oh, yeah. Yeah, we never ran out of power.
Also joining us from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here we had a fresh 2 inches of snow, but it's the fluffy, sparkly kind, so that's nice. And it's currently, where did I write it? Oh, there. 26 Fahrenheit feels like 18, which is -3 Celsius, feels like -6.
So, Kathy, when's the last time you were on a Friday TWiV?
Around New Year's.
That's right. We did our annual thingy, right? Yeah. All right. So for those of you who don't know what I'm talking about, Kathy's on the Tuesday episodes, and we didn't do one this week. So here you are. And from Madison, New Jersey, Brianne Barker.
Hi, it's great to be here. We've got some very light snow, and it's 34 Fahrenheit, 1 Celsius here.
And from Western Massachusetts, Alan Dove.
Good to be here. It's just about the same temperature. It's 2 Celsius, 2 points, or -2 Celsius, sorry. And it's been off and on snowing and overcast and drizzling. And we've got about 2 inches on the ground and some more flurries falling. And it's supposed to just keep kind of dripping through like this for a while.
If you like what we do here on TWiV, consider supporting us, microbe.tv/contribute. We really appreciate all of those who are contributing to our expenses. And if you'd like to join, microbe.tv/contribute. We have a trio of papers to discuss with you today. The first one is I think it's a comment. Yes, it's a comment in Lancet Online. Resurgence of COVID-19 in Manaus, Brazil, despite high seroprevalence. This is something I brought up with Paul Offit last week. I said, why do we have this resurgence? And he said, I don't know. Which is probably what you'll conclude after this as well. But
that's what the paper says.
That's what the paper says. They say there are 4 non-mutually exclusive possible explanations for this. And at the end, they say,
Yeah, we don't know. But I thought we should.
Manaus is, by the way? Do you know where Manaus is?
It's in Brazil.
It's in a remarkable place in Brazil, though. It's not just in Brazil.
In the Amazon.
This is a destination.
Yeah?
Yeah.
It's a big destination. There's a very old opera house there. There's a lovely old Spanish motif. The whole town is that
Have
way.
you been?
And no, I haven't, but I know a lot of people who have, and it's a jumping off place to study ecology in the jungles.
Oh, okay.
It's got lots of destinations after that. So
is it like way inland or?
it's way inland. It's way inland.
Yeah.
So I think the reason maybe for this outbreak is because it's a visiting place for lots of people from around the world.
So Dixon,
check that out, really.
next time there is a debilitating storm in Fort Lee, why don't you go to Manaus?
Well,
Yeah, get out of town.
last time I even considered going into an airplane was quite a while back. And it's been the same. I'm not going to go. Even though I've had my shots and everything else, I'm not going anywhere yet.
Yeah, I'm the same way for commercial airlines. I'm
I
not
want the
getting on an
world to
airliner
recover
till
much better than it is.
till I'm fully vaccinated. That's
Exactly right.
Until the pilots are back in shape.
Yeah,
Right.
that has been an ongoing discussion in the aviation community. So for passenger airlines, for cargo airlines, they're working overtime.
Yeah,
Right.
yeah,
But,
that's true. That's true.
All right, so this is
Dixon, I'm glad you pointed this out. I'm looking at from Google Maps pictures of Manaus. It looks like a beautiful place.
It's in a remarkable place. It's a remarkable city. It's where two major branches of the Amazon River come together.
anyway. This commentary comes from a number of places, including the university in Sao Paulo. There's a hospital in Manaus, and many other places, also Oxford and London and Harvard, San Francisco, etc. But here's the thing. I wanted everyone to hear this because it really kind of says this is what might be happening. So we talked about this study of blood donors, I think, previously, which suggested that this is back in October 2020, about 76% of the population had been infected based on that study. So that's really remarkable. And I think the paper we discussed was about this unmitigated ripping of the virus through the population, right? Basically.
Yes, it was.
So,
Which was, I mean, that was due to Brazil not locking things down and just generally kind of ignoring it. The Brazilian government.
Despite the fact that the president of Brazil actually caught it.
Yes. There are remarkable parallels between there and here. Yes.
You could write a novel on this one.
all right. So,
Well, one of the other things that was really interesting about that paper is that they spent a little bit of time trying to extrapolate that percentage of people who were positive because they tried to account for antibody waning.
Right.
And so that was one feature of the paper, was trying to determine how much antibodies may have waned and sort of adjust their calculations for seropositivity based on that.
yeah, I'm not sure that was accurate, but we'll see. Anyway, so what happened was there was an increase in the number of hospital admissions in Manaus in January, 3,431 through the 19th versus 552 in December 1 through 19. And they actually have a graph here of the different number of events per day over time. So there had been previously an epidemic last year. It peaked in April. Hospitalizations remained stable through November. And they say despite relaxation of control measures during that period. And all right, so what happened? 76% immune or seropositivity, assuming that's correct. It seems kind of strange that you would have this, right? But again, there are 24% of people who are not seropositive, right? And they will argue that they're not mixing, but let's get to that. So the four non-mutually exclusive possible explanations. First is
the data could be bogus.
Yeah, the 76% could be wrong. And then, in fact, the percentage is much lower because they're saying 76% should be herd immunity levels, right? Mm-hmm. Herd immunity against at least hospitalization, I suppose, right?
Yeah, if this is, well, okay, so here's an issue. If this is sterilizing immunity, all these herd immunity calculations are based on complete immunity, right? Like you're not acting as a carrier. Because otherwise the R number doesn't drop to below 1 if you've got 76% of the population that's not going to get sick but can still carry the virus.
Yeah, I think it has to, but they're not testing everyone. So it has to be disease, right? It has to be disease driven.
Right. So they're saying if immunity is completely protective from infection, then 76% would be above the threshold based on the reproduction rate that's been calculated.
That's right.
That's where that number comes from.
I mean,
and that's where they get into the idea that perhaps that extrapolation they did in the previous paper about waning immunity could have been a problem.
Right.
So
then they say, look, if it's in fact lower than 76%, then there's still a lot of susceptible people. And so they're mixed. And that's where the infection has come from, right? But then they kind of try to argue against it. And they say we should have had at least some population protection, right? And not had this bigger outbreak. And they say comparisons of blood donors showed no major difference in demographics. The mandatory exclusion of donors with symptoms is expected to underestimate the true population exposure. So they say, in the end, we don't know. We have to look at the data again. So, okay, that's the first part. That number may be wrong.
Right. Might not be 76%.
Which I think is a likely possibility. Okay.
Sure.
That seems damn high. And then the second is that immunity against infection, they specifically say infection, right? Not disease, might have begun to wane by December 2020. And, this is certainly happening. And they cite a few studies in the UK. Reinfection is uncommon until 6 months after the primary infection. But most of the infections in Manaus occurred 7 to 8 months before the resurgence in January, which is longer than the period in the UK study. But they say, nonetheless, waning immunity alone is unlikely to fully explain this resurgence. Again, it depends what you're looking for. If you're just looking for infection, and I doubt they are, right? They're looking at hospitalization.
Yeah, they're looking at hospitalization. So these are not just symptomatic cases. These are bad cases.
And I would think that even if you have waning immunity, which is normal, your antibody levels should go down, you're still going to be protected against severe disease because you're going to have some kind of a memory response. It may not be quick enough to prevent infection, as Paul Binesh had suggested a couple of weeks ago, but it should prevent serious disease.
Right. So the other thing that I didn't mention before when I was talking about Manaus was that it's a major commercial exchange center. So I'm curious as to how many of these people who got sick were actually residents of Manaus or had just gotten sick and gone to Manaus for treatment.
Mm-hmm. Good question. I don't think
they do comment that apparently there are some data showing the population mobility has gone down. From November to December in the area.
Yeah, but down in there is
right. Your point still stands. There can still be a lot of exchange from other areas where people were not as exposed.
I mean, almost everything from the draining basins of both major bifurcations of the Amazon River end up in Manaus. So it's a wide open question.
Yeah. Okay. All right. Third, then they bring up the issue of the variants with antigenic changes, which may impact the ability of these viruses to reproduce in the presence of previously acquired antibodies, right? So they're these three lineages, B117, B1351, and P1, have various changes in the spike, as we've talked about before. And one of them has been detected, P1 has been detected in Manaus on January 12th. But that seems, one of them has been, I don't know what fraction of these cases are with this.
My impression was that they don't really have sufficient data to address that hypothesis.
I agree. Yeah, see, they say one case of reinfection was associated with the P1 lineage that had 10 spike protein changes. But I have a feeling they're not doing enough sequencing, right?
Yeah.
Where does P1 come from again?
P1 is a Brazilian variant.
Oh, it's a native. Okay.
First observed in Brazil.
First observed in Brazil.
Right.
Yeah, who knows where it actually.
Who knows where it came
Yeah,
from.
and we can't totally blame them for not doing enough sequencing because I'm not sure how many places are doing.
No,
it.
I'm not blaming them.
Nobody's doing enough.
That's exactly right.
I'm not blaming them. I'm just saying that could explain why we
That
don't
would
know
explain
the answer.
why the data aren't there.
And now they have another lineage, P2, which is a sublineage of B1128, has been detected in several places, including Manaus. So, and as we have said before, some of these spike changes can reduce neutralization by antibodies, both monoclonal and polyclonal. So, we
still don't know what the implication of that is for
this
actual immunity, but the laboratory observation is that they do seem to.
Yeah.
Right.
Now, I mean, one of the reasons I wanted to bring this up is because, the mainstream media says this is all variants doing this, it's driving this. And I don't think it's right to conclude that. I mean,
wait, do they sequence a lot of stuff, by the way?
No.
Great.
Mainstream media?
Oh, I see. They don't do any sequencing.
No, they don't.
The journalism toolkit does not come with any sequencing equipment. I'm sorry to report.
Not even in the field.
No.
All right. And fourth, SARS-CoV-2 lineages in the second wave might have higher inherent transmissibility than before. And so they say the P1 lineage reached a high frequency among genome samples, 42%, 13 of 31 of genome samples obtained from COVID cases in December, but was absent in 26 samples collected between March and November. And they say, we don't know anything about the transmissibility of P1, but, it has some changes with the other variants, which have been suggested to have increased transmissibility, allegedly increased, right? So we don't know. They say, we basically don't know, but it's a possibility, and that's fine. But, the problem is that any of these things, if any one person talks to a journalist, that's what they'll pick up, and that's what goes forward, right?
Absolutely.
So the bottom line here is that we don't know what's driving this outbreak. And
this is why if you are one of these authors and you're talking to journalists, you need to lead with, well, there are 4 different possibilities here, and we don't know which, if any, of them is going on.
Good.
4
Yes.
different possibilities.
And they're not mutually exclusive.
No, they're not.
at all.
No, they are.
It could be a combination of all of
Could
these.
very well be a combination.
non-mutually exclusive, right?
And I think we're not going to get answers on this unless we have the misfortune of seeing similar patterns elsewhere.
Yeah, you may be right.
I mean,
right.
Because this could be some fluke of local conditions. Dixon was talking about the traffic flows through the area. Every city is unique.
Agreed.
It could be the timing of their first wave allowed immunity to wane just enough, and then some of the variants had just enough escape, and it could be a combination of factors. Or this could be a common theme that shows up again, but we haven't seen it elsewhere yet.
So is this
a lot of places we're seeing continuing decline.
Is it possible also that it's some healthcare worker at one of the major hospitals in Manaus? I wouldn't know how many hospitals they have altogether, but they can't have many. It's not that populated.
One super spreader event?
Yeah.
I don't see why not, right?
Right. So that's number 5.
So, Alan, the thing is, Alan, what you said is right. If you talk to the press, you say there are 4 possible, but at the end, they're always going to say, but which one do you think is the most likely?
Yes.
Yeah, that's true.
Right? And not everyone can resist answering that, right?
I think the most likely explanation is all 4.
No, pick number 6.
Pick number 6.
Exactly. All right. So that, I think, everyone needs to understand this, that we don't really know what's going on there. So, but the press will sometimes conclude, and I think we have to keep our minds open.
Yeah.
It looks like there are 3 hospitals in Manaus, according to Google.
You've never been there, Kathy, right? Because you've been to Brazil a couple times, right?
Yeah. No, just one time, but I did not go to Manaus.
Okay.
I think it's interesting.
Population 2 million. I looked it up.
2 million.
Good sized city.
More than I thought. That is more than I thought.
I think it's good that sort of this interesting phenomenon is being reported on. And I like that they talk about it as, 4 possibilities to help us learn about more. But, that's as much as we can really say at this point.
Yeah, it bears watching.
All right, our next little story here is an article in Nature Communications. Evidence for SARS-CoV-2 related coronaviruses circulating in bats and pangolins in Southeast Asia. Okay, Southeast Asia is not China, folks, just so you know.
Let's be clear about that.
I'm amazed that there's a pangolin left in Southeast Asia.
These are from last year and before. So they're not.
Well, and the ones they sampled were confiscated, weren't
Yes,
they?
these are all confiscated from
Somebody
illegal
was smuggling them.
traders, and they don't even know where they came from. So
Right.
anyway, this comes from a large group in many countries, including Thailand, of course, but also the Singapore. Lin Fa Wang is the last author on this paper. Thailand, Australia, China, Virginia, Singapore, and these authors contributed the Wakara Pluesadi, Tan, and Maniorn. And both Linfa and Hema Chuda jointly supervised the work. Those are the last two authors. So as everyone knows, SARS-CoV-2 likely originated from a bat somewhere. We don't actually know where. And what was it that, this bears repeating, because I don't know who said this once, but where did HIV/AIDS start compared to where we first detected it?
San
this-
Francisco or
The story
Close.
is
Right.
it originated in Cameroon.
Yeah.
And it was first detected in a group in Los Angeles. I think it was in the first
That's right.
paper.
That's right. So
Can you tell I teach a first-year seminar and cover these things?
yeah, it's good. So that's, where viruses emerge from wildlife is not often where you first see them. Okay, folks? So here, this is part of an effort
That's why we keep trying to back off from things like the UK variant, the South African variant.
Yeah.
And it's,
Well, today we did well. We did B11.
Yeah, we did okay on that.
And
Yeah.
P1.
And the thing is that bats, in general, pangolins don't migrate much, but bats
go
are one of the
everywhere.
biggest migrators.
What's the migration range of a bat, Dixon?
All over the place.
Depends on the bat.
That
Yeah,
is
it
so
depends on the bat
precise.
species.
That's very precise.
Oh, you'd have to ask Peter Daszak. He knows all about that.
Well, I think some are
Depends on
limited.
who's swinging it.
Some go long distances, but some
Good.
do not.
I
In
like
fact, there's the NEPA paper we did on bat. Remember, some of those bats stay in a very local area, and others go farther. So not all of them go all over the place. Dixon, I don't think a New York bat is going to end up in Thailand, is it?
Only if they get traded.
If they get find their way into a shipping container.
Right.
Yeah, that's
We've
true.
had a number of cases of wildlife that got transported.
Or
that
if
way.
it's,
Maybe-
confiscated by wildlife
Yes.
bandits.
maybe on the order of 2,000 kilometers.
Bingo.
Okay, that sounds reasonable. So
2,000.
Kathy, am I 2,000 kilometers from you?
Oh,
You're probably less than that.
Yeah.
2,000.
More like 500 miles.
Let's see,
so
New York
less.
to Ann Arbor
That's going to
in
be
kilometers.
like 1,300 kilometers or something.
Yeah.
Oh, the air distance is 500 kilometers.
Right,
I'm
500.
sorry, I
Oh,
don't mean to put you on the
right.
spot, but I thought it would be a good illustration. So it must be
Sure.
more like California, New York, right?
A little bit. So that's 3,000 miles.
That's
a little bit
like
more.
7,000
Oh, that's more.
kilometers or
Okay.
something.
Right, well, it's like
Anyway, so the idea is that SARS-CoV-2 probably originated somewhere in a bat, but we don't know where. And the reason we say that is because a virus called RATG13 has 96% whole genome sequence identity, but that was identified in 2013, 1,000 kilometers outside of Wuhan. And I just want to remember everyone that we looked for the ancestor of SARS-CoV-1. Not we, I didn't, but we as the scientist community. I don't do anything. And they found it in a civet, and it's 99% genome identity. So that's what you're looking for. And then you can say, this is it. So they're looking, and this group has looked, and that's
Right.
what we're
So
going to...
RATG13 is almost certainly not the immediate ancestor of SARS-CoV-2.
No, that and SARS-CoV-2 share an ancestor.
Related closely to whatever was the immediate ancestor, and it's in bats.
Yes, and
as a matter of fact, we did a paper on that some time ago that predicts when they diverged, and it was decades ago.
Yeah, that's right.
Yeah,
that's right.
And one thing for people to remember as well, this is part of a little bit of figure 1 of the paper, which I found really helpful. RATG13 was described in a part of China that it was very far away from Wuhan.
2,000 kilometers. Yep.
Yes. And so it's nice to kind of, for me, seeing it on the map made it very clear.
All right. So this paper is about a sampling in Chachoengsao province. Probably I massacred that. Thailand, which is a colony. This is great. I love this. A colony of 300 bats in an artificial cave made by a 1 meter diameter irrigation pipe in a wildlife sanctuary. This is in the east.
Nature abhors a vacuum, Vincent.
So you make a space and things will come. Yeah.
That's exactly right. Exactly right.
Well, like the bridge in Austin, the bats live in the bridge, right?
Yep.
Or like the George Washington Bridge and the Peregrine Falcons.
Or buildings that humans build and humans live in them, right?
Keep going. No,
it's not the same.
If you build it, they will come.
If you build it, they will come. That's how to say it. So
if you don't build it, they'll still come.
They captured 100 bats and did rectal swabs. By the way, there's only one species of bat, Rhinolophus acuminatus.
And this is this location, Chachoengsao, is all the way at the southern end of Thailand. So Thailand and China don't share a border; they're close. But,
yeah, that's right.
So if you went from the southern southwest corner of China and then some distance into Thailand, you'd have to cross all of Thailand to get. This is probably not a bat that migrates up to China.
Right.
No, although the place where RATG13 was described was sort of in the part of China that is closest to Thailand.
That is closest to Thailand. It is the southern part of China.
Yunnan,
as I'm reading this, I'm imagining that it's convenient to get at these bats because they're in a in a 3 meter diameter drainage pipe. There's 300 of them, and I noticed that they sample 100 of them. That's 30% of the whole population. Yeah. You probably just stick a net over the end of the pipe.
They did a reach in there and grab bats. Yeah,
they'll bite your fingers. Watch it.
All right, so they sampled 100 bats, and then they do PCR. They have a pan-coronavirus PCR for a part of the RNA polymerase gene. And by that, I say 13 of these 100 rectal swabs are positive, and they sequence what they amplified, and they're the same. All of these are the same, and they're 96% identical to RATG13. And notice I said 96% identical, not homologous. I learned, right, from people telling us.
Right. I remember the letter.
Identity, sequence identity. And the authors have got it too. And it's a 95.86% identical to SARS-CoV-2. It's almost as good as RATG13.
So it's further from SARS-CoV-2 than RATG13, but very close.
And there's another very close bat coronavirus isolate that's been discussed. We have discussed RMYN02, 94.48% sequence identity. All right, so then these are PCR products. They then say, let's sequence the whole genome. So they do whole genome sequencing, and they get 5 whole genomes, and they say basically they're all the same. It looks like there's just this one virus in this whole colony. So it's the same species of bats, and they all seem to have the same virus, which they name after me, RAC-CS203. Just kidding, folks. RAC, then a capital C, capital S, 203, which RAC, of course, is Rhinolophus acuminatus. So I guess I'm a bat in the end because I'm Racaniello. And say this is the most dominant coronavirus circulating in this bat colony at the time of sampling. So that's pretty cool that they go into this pipe and they find something which is really close, which tells me that you could go almost anywhere and find
There are
closely
always more
related.
viruses in the pipeline. Yeah.
So, isn't it unusual, Vincent? Don't you think it's unusual that you have a clonal species of virus without change? Absolutely no difference.
Well, I don't
in
think there are
100
no change, but they're very similar, right? Almost identical, they say.
Well, if you took a
Almost
population of humans,
That's not like almost pregnant, is it?
no. If you took a population of humans who were infected with flu and you sequenced all the viruses from them, you'd find a lot of exactly the same strain of flu, right? So it's not that unusual to find the same strain of virus in this
I don't know.
close-knit group of bats.
So look, in humans, any two isolates of SARS-CoV-2 up until recently were 10 base different out of 30,000, right?
That's incredible.
So maybe that's what they're talking about here.
Okay.
Yeah,
10
and they
10
only did the full sequence of 5 samples.
Yeah. So I think that's close enough. Yeah.
And they say that they're almost identical.
Yeah.
So there's going to be some variation.
I think
Couple
10
of
out of
nucleotides,
30,000 is almost, right?
Yeah.
That's
Right.
pretty good. All right. So we have this virus now, which is quite similar. And now they have a new, they say, okay, this virus, and they do phylogenetic analysis. They say this is a new member of the SARS-CoV-2 related coronavirus lineage, which is called SC2R-CoV, SARS-CoV-2 related coronavirus lineage. And that's different from the SARS-CoV-1 related coronavirus lineage now, right? 2 separate lineages. We have representatives of both in humans and also in bats. So that's what this is here, and but it's damn close to SARS-CoV-2, and so there are probably many more out there. So then they do some interesting work. They take a look at the receptor binding domain. So let's see if we can figure out what receptor this spike is binding to, right? And they say, we look at it, phylogenetic analysis, some structural analysis. It doesn't look like it's going to bind ACE2. And in fact, they make the receptor binding domain protein. They produce it in cells, they purify it, and they do binding analyses with ACE2. And doesn't look like it's binding ACE2. And then they also make a VSV pseudotype virus with this in it, and it doesn't infect vero cells, which can be infected with SARS-CoV-2 via ACE2. And the last sentence is quite interesting. As the sampling was done using lysis buffer, no virus isolation was conducted. However, we will be attempting virus isolation in future sampling studies. And you do have to get virus at some point, right? Because
They're saying, Vincent, don't get on our case about this.
I wouldn't. But they're saying that so that the gain of function people don't go after them, I think.
That too.
So it's not binding ACE despite being very close, right? In fact,
close enough.
not close enough. One amino acid is all it takes, of course. One bite is all it takes. One amino acid is all it takes. And so forth.
Remind me, I'm blanking. Does RATG13 bind ACE2?
Don't believe it does. That's a good, we can't keep all these facts in our head.
Yeah.
I don't think it does.
I don't think so either.
My recollection was one of the reasons that the pangolin stood out is that
Identical.
virus.
Right.
That's right. That's right.
That virus had an ACE2
It does.
receptor binding domain.
Oh.
Yes.
Right. And so they were wondering, I remember early on, they were wondering whether the pangolin was an intermediate host and the RATG13 picked up, the appropriate ACE2 specificity through some recombination or something in the pangolin, some sort of story.
So it does bind ACE2. RATG spike does bind
it does
ACE2.
Bind ACE2.
I had it wrong. Okay.
Yeah, we did have it
So
wrong.
maybe it's the furin site I'm thinking of that was in the pangolin.
There's certainly no furin site in RATG13.
It's
Yeah.
always an alternate explanation for everything.
I feel like my recollection was the same as Rich's. So there's something that was especially
There was something that was particularly attractive about the pangolin
Yes,
model. Yeah.
right.
Even though the virus they actually isolated from the pangolin was less identical than RATG13, but
The pangolin isolate also binds ACE2, and I believe it has the furin cleavage site. Yes. All right. So a little bit of serology. to see, what's the extent of circulation of this virus. And they have an interesting assay, which they call a surrogate virus neutralization test. And basically, they're asking if antibodies can block the receptor binding domain binding to ACE2. Okay. And that is a surrogate because not all the neutralizing antibodies are going to do that, right? A fraction of them will, but some of them will neutralize by other mechanisms. But this is an okay surrogate. And so they have serum samples from these bats. I presume they're from this bat collection.
Yeah,
Right?
that bugged me too. They do say 98 serum samples, and that's pretty close to 100. But I read the first 3 lines, and I was thinking, are they collecting these from humans in the area?
Yeah,
They didn't talk about that. They never said bat serum.
No, and they didn't say specifically here if it was their selection, their collection of samples, because, if they start off by saying we're going to investigate the circulation of this virus or related viruses in Southeast Asia, but then they only look at their own. So
In this one pipe.
in one
Right.
pipe.
In this one pipe is Southeast Asia.
Anyway, of these 98 samples, 4 had antibodies that could block RBD ACE2 interaction, and 2 of them very strongly, 88 and 97%. All right. So that's their little study. So that means that, 4 out of their 98 bats were infected at some point. And they also tested 10 pangolin sera, which were collected last year, February to July, at 3 wildlife checkpoint stations in central and southern Thailand. And they don't know where these animals came from because they were captured from illegal traders, right? So they're in, don't pangolins come from Malaysia a lot, often?
All over the place.
All over?
They're all over the
From
place.
Fort Lee, too?
I used to have one as a pet, yes. I can show you. I have a model of a pangolin.
All right,
There's many species.
Anyway, they confirmed that these were actually pangolins, which is, by the way, folks, if you have a serum sample, you could sequence mitochondrial genes and confirm whatever species. That's what they did with the bats, too. And these are Manis javanica.
They weren't just some other animal disguised as pangolins being smuggled through.
Well, if someone gives you
Someone gives you serum you want to check to make
you
sure it's
should
what they said
probably-
it was. Yes.
Yeah.
Exactly right.
you should probably check
Sorry, Vincent, what was the species name on that pangolin?
Manis javanica,
Java.
All right, so they say
That's probably where
that's
that
an
came
abundant pangolin species. All right, one sample had a strong so again, there's this surrogate neutralization assay, 91%. And they say we have historic data in China from May of 2003 showing 1 of 7 pangolins here are positive for SARS-CoV by both ELISA and virus neutralization. And those pangolins from 2003 were quarantined at the Guangdong Wildlife Conservation Center, again, taking them from illegal traders. And those come into China from Southeast Asia. Again, we don't know what country. But basically 2 separate studies where, pangolins are susceptible to infection by SARS-CoV-2 related coronaviruses, because their assay in this paper is with SARS-CoV-2 related coronaviruses.
And the 2003 was SARS-CoV-1.
Right.
That's right. Because, of course, we didn't know anything about SARS-CoV-2 at the time.
If only we had.
Only we had, indeed.
Wish we had known.
Then they do a study, which is I find a little weird, but so they say, this following study is not actually done with RACCS203, which is the virus they identified in their pipeline.
In their pipeline.
But they used RMYN02 receptor binding domain, which they say is only 2 amino acids different. So we're now going to use that as a surrogate for RACS203. I thought that a little weird, right? But they made the recombinant protein, RBD, they tagged it so they can track it through their assays. And then they asked, do patient sera react with different receptor binding domains? of these various viruses, but they didn't check RACCS203. They checked RMYNO2, again, which is 2 amino acids different. I don't know. If it were me, it's easy to make these proteins. I would have done it. Because isn't it kind of weird to say we're using it as a surrogate?
There must have been some reason for this.
Because 2 amino acids could make a big difference.
Sure.
Anyway, so the strongest reactors with human patient sera, people who have had COVID-19, the strongest reactors are SARS-CoV-2. Makes sense. That's what infected them.
Good. Yeah.
RATG13, which is very close, and then GXP5L, and I don't actually know what that is. It must be another bad isolate. And then the weak reactors, ZC45 and RMYNO2. And so they're saying, if we had done this with RACCS203, it probably would have been a weak reactor because it's only 2 amino acids different from RMYNO2. And finally, SARS-CoV was also a weak reactor with these COVID-19 patient sera. And they also did this with SARS-CoV-1 patient sera, and they had low cross-reactivity against all the SARS-CoV-2 RBDs. And that's what you would expect because these are now 2 lineages, right? And they're quite different. Okay, so what does all this mean is that they're related. These RBDs are somewhat related, and you can have cross-reactions. And then finally, they made rabbit antibodies against different RBDs. And then they basically say they have the same pattern as they already observed from the human sera, cross-neutralization.
And they have a map. They have a nice map in Figure 4 of the distribution of these different bat species.
Yep.
So the one that they got the CS203 from is a southern Thailand species.
Yeah.
And then the RATG13 is found in a species that actually does extend into northern Thailand, but southern China and northern Thailand.
Right. And then they actually talk about some other SARS-like coronaviruses that have been isolated. And of course, Thailand, China, and also in Japan, apparently, in the north of Japan, one has been identified. And so Iwata Prefecture, Japan in the north, SARS-CoV-2 related coronaviruses in bats. I didn't know it was in Japan. Did anyone know that?
No, that was news to me.
So this is, they say, 4,800 kilometers. I wouldn't ask anyone to give me it's not like New York, Austin, right?
It's long.
I always just kind of divide by 2 and then add a little bit so it's like, I don't know.
Oh, here's one. New York is 4,500 kilometers north of the equator.
Ah, okay.
All right, tell me what country.
I was going to say like 2,500 miles or something.
Where does the equator pass through on this?
Middle of the earth.
No. In this hemisphere, what country does it pass through?
Brazil.
Ecuador.
Okay. Got it. Now I got that. So 4,800 kilometers, we have SARS-CoV-2 related coronaviruses from bats. That's a long ways.
A long ways.
I told you bats migrate far.
Well, so...
No, I don't know they go there.
One of the things I'm wondering about here is people are looking and looking and looking at these coronaviruses in bats in all these Asian countries. How thoroughly have we looked for coronaviruses in bats in any other region in the world?
Ask Peter Daszak.
Yeah, I actually remember that came up I don't remember if it was Peter Daszak or Tony Shounts or both or somewhere, but I remember getting the distinct impression that the idea of the bats in, say, North and South America had not been really surveyed at all for coronavirus.
They'll say that at that point we ran out of money.
Right. Right. So just so people don't get the impression that this is isolated to, an area. I mean, now we're seeing it from northern parts of Japan all the way down to Thailand, which is this huge swath, and people just haven't maybe looked closely enough anywhere else in the world to see all the coronaviruses that are circulating.
So who wants to Google and find out how many species of bats there are?
Oh, too many?
Yeah,
Isn't it
20%
20% of them.
20% of mammals
It's
are
a lot.
bats.
Yeah, but how many mammals are there?
Well, I don't
not
know.
as many as beetles.
It's a very no. That's true. That is true.
Dixon,
There's a lot.
is a camel a mammal?
A camel is a mammal.
What was a mammal?
That's a book, right? But wasn't that a book by Dr. Seuss?
It's a camel, a mammal.
All right, let me Google it. Okay. Is a camel a mammal?
1,400 species of bats.
Yeah,
1,400.
that was Dr. Seuss.
Okay.
I'll be darned.
1,400
No, I'm
is a
sorry.
doable number, though, right?
It's not by Dr. Seuss. It's
Yeah, I
by
don't think so.
Tish Robb.
Ah,
I don't know who that is.
good. Because if it was Dr. Seuss
Sorry.
and I didn't know it, I would feel bad since I live in the
Yeah.
Springfield area.
So basically, they say it's safe to predict that many more SARS-CoV-2 related coronas will be discovered in bats once surveillance studies are intensified through broader and well-coordinated international collaborations.
Right.
And that's true. We haven't sampled enough. But what I think
Question
is yeah, go ahead, Dixon.
No, I was just going to ask if museum specimens would be a good source for at least trying to find the answers, because if we know there are 1,400 species of bats, it's because we collected them.
What would you expect to find in a desiccated bat?
No. It's preserved in alcohol.
Well, they're
Formaldehyde.
yeah. So I've
That
actually
was a question. It wasn't an answer.
no.
Kath.
It's a very good question because I've talked to people who are doing this work, and it turns out that you can get usable sequence out of those animals. People are generally looking at the animal itself, like sequencing the DNA to find lineages of museum specimens.
Exactly.
It is very, very challenging. There's a tremendous amount of degradation of the DNA. If it's been preserved in formaldehyde, it's actually worse than if it's just been dried out. And I don't think anybody has really tried to get RNA out of those. I'd be very surprised if you got usable viral RNA sequence out of a museum sample that wasn't a contaminant. But it's a neat idea, because if you could do that, then you could maybe get a handle on it.
Exactly.
And in this study, they had the pangolin sera, and they weren't able to get any nucleotide sequence out of that.
Yeah.
Right.
Yeah. Which is presumably kept frozen, right? So
Yeah.
but although it may have been collected after the infection was over.
Well, and it's serum, so it's cell-free, right?
Yeah. I think it's worth looking at, but I'm not sure.
It's definitely worth trying.
But there's certainly DNA
the museum
in
specimens
those.
would be a lot more accessible.
There's certainly DNA in those because people have recovered, endogenous retroviruses from museum pelts and so forth. But RNA is a different beast. Anyway, the one thing I think that to conclude this is that now we have a big range for SARS-CoV-2 related coronaviruses in bats, not just in Wuhan area. So it's really not a good idea to blame the Wuhan lab for this virus because 4,800 kilometers, it could be anywhere in that, and probably even a bigger range too, right?
Yeah.
So
well, and it's not even necessarily true that the virus originated in China.
Yeah, that's right.
It probably did because that's where you see the first cases. And even when you go back to blood samples, you see the very early cases before it got to the seafood market. Those are all in China. So that is most likely where the spillover event happened.
I agree, but we don't know. Maybe not.
I mean, and you've got these bats migrating across borders, and so there's a lot of possibility there.
I mean, for
And
SARS,
like Manaus, Wuhan has a lot of human traffic going in and out.
A lot of people going in and out, or at least they did.
I mean,
but there's a lot less right at the moment.
I think we agree that SARS-1 originated in Guangdong, right?
Guangdong, right.
And then they went to the countryside and they found 99% identical viruses in civets. So that's pretty good, but we don't have that
MERS?
yet.
MERS?
No, that's SARS-1.
No, I know. And MERS? Where did that originate?
Well, that's a good question. It's mainly where there are camels in the Arabian in the Arabian Peninsula. Yes, right. In countries in the Arabian Peninsula, that's where most of the cases have originated. They've been exported to a few other countries, but they originate there. And,
but we now know the virus is pretty widespread in camels, right?
Yeah, many camels.
Yes.
The right camel, right? You have to have the right number of humps, and I don't remember what it is, but
One.
One hump?
I think it's, I think
Two
they're
humps
one
is
hump.
very Bactrian, the Bactrian camel.
Yeah, the one hump is dromedaries.
dromedaries.
Yeah. Okay. So that's what
there
it's
are
in.
more camels in Australia than there are in Arabian Peninsula.
Ah, so
And
that's.
the-
Go ahead.
No, mine was a trivia fact.
That's a trivia fact.
was a trivia fact.
That was very trivial.
just so how you can remember which is which. The dromedaries
Is a
are like
D.
a D, so think
Yes.
of a D, and the Bactrian
is a
up
B.
to are like a B.
Assuming
Yeah,
you
well-
want to know, assuming you want to remember, right?
And if you want to see a lot of Bactrian camels, go to Tibet and go to Mongolia. They're all over the place.
No, the point you made, Dixon, the camels in Australia, are they dromedaries?
Yes, they're derived from Arabia.
I wonder if people have looked for antibodies.
No, that's a great question, because the way Australia was explored was that the explorers, and I'm blocking on their name right now, heirs, took camels from Saudi Arabia and went from Adelaide all the way to Darwin. And then once they finished their expedition, they released them.
Because what could go wrong
And the rest
releasing
is
a species
history.
in Australia?
The rest is history. Well, they've been doing that forever in Australia, so why stop now? So that you can see camels everywhere in Australia. They're just everywhere. It's a perfect country for them.
So the other takeaway that I get from this paper is that there are lots of SARS-1 and SARS-2 related viruses out there.
Yep.
Yes.
Okay.
Yes.
So the potential for spillover is enormous.
So watch this space. The next epidemic is due any minute.
And I wonder, this is way down the road, but, surveillance for spillover is an interesting concept.
Yes.
And I wonder, people who are smarter than me are probably analyzing all of these genomes and trying to understand whether there are any sort of signatures that you can see that suggest whether something has more potential than something else for spilling over. That's going to be part of the surveillance.
Right. And I think that, in our conversation already, we've seen multiple examples of places we ought to be looking. There are lots of these viruses, and we still have this long list of places to look.
I want to show you guys this paper, MERS corona antibodies in Bactrian hybrid camels from Dubai.
I didn't know they interbred.
But, here's a map of the world that shows you the geographical distribution of dromedaries and Bactrians.
Right.
So the dromedaries are yellow, right? And the Bactrians are green.
These are single-humped camels.
So they're yellow, and so Australia has both, apparently.
Both. Yeah, they have both.
And when you see a little camel, those are seropositive animals. So both Bactrians and dromedaries. But I don't see any icons in Australia, but they may not have looked. I just don't know.
Right.
No, and the good thing is that the camels in Australia are exported. They don't import any camels. So it will all be one way.
It's interesting.
Right. So in terms of the origin of MERS, we don't really have a handle on where it got into camels.
No.
Right.
No.
And now it's widespread among camels in North Africa, the Middle East. And so you have these spillover events that happen there.
We not only don't know from whence it came into camels, but when.
Yeah,
It's possibly been in camels for a long, long time.
It's possibly been spilling over into humans for a long, long time, and we only just noticed.
Yep. Okay. I think that's very interesting work there. I think we need more of that.
Do you see how many subjects we just covered? This isn't just about virology, folks. This is about life in general, because you can't know anything about life in general unless you learn about viruses.
Viruses are the world.
Everywhere. They take you everywhere.
That's the thing about having a chat, right? You can wander in different areas.
That's right. Some people criticize this podcast for that reason. I think the reason why I'm still here is because of that.
Because you chat?
Well, because I don't know much virology, but I know a lot of other things, I guess. But, these are nickel.
You make conversations more interesting, Dixon.
Well, and you make them funnier.
All right. And we have one more brief study, which is a preprint. This is decreased SARS-CoV-2 viral load following vaccination. So we are all trying to figure out if, and I use we again in the grand sense, whether immunization or vaccination with the COVID-19 vaccine will impact shedding. So,
and again, as a preprint, we apply the usual asterisk. This hasn't been through peer review. It's not done yet.
Yeah, so this is, in Israel, they're doing a good job of vaccinating many people.
Yes.
So this comes out of a number of institutions in Israel, the Israel Institute of Technology, Maccabi Health Services, Tel Aviv University. And we have Levine, Tiefenbrunn, and Yellen, first two co-authors. And then we have Kishoni as the last author here. So this is interesting. As of January 25th, Maccabi Healthcare Services vaccinated over 650,000 of its members as part of the national rollout. And these, apparently they do SARS-CoV-2 tests in their laboratory. So they said we could track post-vaccination infections. I'm not quite sure what is going on there, right? I guess, the members of this health service have had SARS-CoV-2 tests regularly. And so,
right. So I'm assuming Maccabi Healthcare Services is like a Kaiser Permanente sort of big organization that has all these patient records. And so they're doing a lot of the testings being done in their lab and the vaccinations being done through their clinics. So they have data on people who've been vaccinated, and they have follow-up data on people who've tested positive before and after.
Yeah, one thing that's interesting that they don't really talk about a lot here is, the proportion of those who are vaccinated who get infected versus those who don't get infected. And so that question of protection from infection is not really discussed here at all.
Right. And I'm not sure that they even could get reliable numbers on that from their data, because it would depend on how much of the country their data set encompasses. So you might end up with weird skews in it.
All right. So basically this health service is doing PCR tests, and they have data which they collected after it's already been done, PCR tests, with multiple viral gene targets. And they can know of those who's been immunized, vaccinated when, right? And they can take that data and look at how much, what the CT value is essentially.
Did they actually say in here how many patients were positive out of this group after vaccination? I'm looking for a number and I'm not finding.
Yeah, there's
vaccination positive tests.
Yes.
Yeah. So they've got.
Yeah, they don't really.
Well, they've got it in different groups.
Well, they say here, comparing post-vaccination positive tests from days 1 through 11, the N is 1,755.
Okay. Right. And days 12 to 28 is 1,142.
Right. Those are the numbers. So they look at the CT values from these PCR tests in days 1 to 11 or days 12 to 28. And they say, what's the CT value after vaccination, right? And what is the CT value?
And they've got a demographically matched unvaccinated control group that they're going to compare to.
That's right. They controlled for age, sex, vaccination, et cetera. So basically what happens here is that, all right, so a difference of one CT unit, they say, is equivalent to a factor of 1.94 in viral particles per sample. So the differences that they observe represent a viral load ratio ranging from 2.96 to 4.68. And last night on TWiV, Daniel said this difference in CT, which is here, the differences ranged from, 1.6 to 2.33 for vaccination after 12 days, and compared with, what's the CT on the other? I can't find it. It's you can look in the picture, actually. It gives a nice graphic view. Daniel said this difference in CT value, which, let me put the picture up, corresponds to a drop of 1.4 million RNA copies to 400,000 RNA copies. So let me put up this. And for those of you who are listening, we will try and describe this this graph here where it's...
And this is open access.
It is open access.
Preprint that's...
Yeah, you could pull over and get it up on your phone. Right. Yeah. The y-axis is CT values, which range from 23 to 30. And the x-axis is days after first vaccine dose. And you can see from days up until day 7, 8, 9, 10, 11, this CT is hovering around, almost 25, right? With some variation. Yeah. And then it goes up day 12 with
right. Higher CT value is less virus.
It's 27 now, right?
Yeah.
Mm-hmm. And, the error bars on these are big, but some of them are above this line that they've calculated here. So it's going up in 28, 29, and so forth. Okay, that's the finding. So what do we think about this?
So you said, according to Daniel, it was from 1.4 times 10 to the 6th to 400,000.
Yeah.
That's pretty impressive.
Because that's the question. Does that change so there's some impact on CT in this later time of after vaccination. The question is whether that would impact shedding or not, right?
That's the big question.
What's missing is how many sneezes or viral loads. What is your viral load that you need to transmit this thing at? These numbers don't mean a thing unless you know that.
All you know is there's a difference, right?
And they were assaying the RDRP, N, and E.
Right.
What I wanted to know is, okay, this is RNA. What about infectious virus?
Yeah, of course. So these are,
I
mean, these are stored data that they don't.
It's right. Data of convenience.
Yeah,
right.
Yeah. No, absolutely. Sure. That has to be done. But
so I was trying to figure out: are any of these in that set of genes that are overrepresented because of the nested RNAs as opposed to virus? I'm thinking back to what Susan Weiss.
These are all going to be the PCR products that are in whatever standard test is being used in Israel.
So the RDRP, of course, is the whole genome, right? Only. And then E and N are the subgenomic mRNAs, and they're going to be both. They're going to overlap. The mRNAs are going to have both sequences.
Right.
So at CT levels, what is considered negative on this one? 34.
Negative in terms of what? Having
no virus. It's
0?
Just random. Yeah.
Well, you
showed 2 before.
Yeah.
Right? So how many cycles do you go to before you say, no, that's not
before you say it's not there?
Yeah.
Well, these are real-time assays where you're measuring fluorescence, right? And then when it's above background, that's the CT. So, I mean, I think
when Vincent at his CT value and it was 35, he said, or 37.
Exactly.
Yeah.
It was
random event.
Yeah.
Yeah, I think 37. The problem is, though, different machines and different primer sets can give you slightly different CT values with the same sample, right? So it's really hard to say at this value you're negative.
Right. So then Kathy's point is even more well taken then because...
Yeah, I agree. I think we need to do infectious virus.
Sure.
But this is kind of a hint, right?
Yep.
So this is taking data that they had and saying, can we get a first pass at what effect the vaccine might be having? And it would have been pretty eye-popping if they'd seen no effect, right? I mean, if it was all just, the virus is still at the same levels when you get reinfected post-vaccination, that would be worth comment. And here, okay, we see reduced CT values pretty consistently, and there's variation, but pretty consistently the post-vaccine group is having lower CT values than the unvaccinated group, and lower CT values after 12 days. That's a pretty strong indicator that there's something going on to suppress the virus.
Right.
That's the thing that I find most interesting is this apparent, almost really pretty abrupt break.
It's dramatic. Yeah.
Between and around 12, 13 days.
Yes.
From lower CT values to higher CT values. The lines they've drawn through them are a little distracting.
Yes.
It would be nice to see some sort of linear regression analysis.
Non-statistical lines. Yes.
Yeah. Well, except for this one patient.
Yes, there's one patient who's in between. Yes.
Can anybody here imagine a device which you breathe into and it tells you how many virus particles you're shedding?
I can imagine it. I couldn't.
No, but does one exist?
No.
Actually, there is. I saw back in the day when we were all about testing and everybody had a new idea, was somebody for a while had some sort of machine that would assess particles in breath.
Mm-hmm. Yes.
And had some way of counting virus particles. It was
it was a density.
They were going to use it at Heathrow or something like that.
This was an MIT group, I think. No?
So I forget. But I remember seeing one report and no follow-up, which is probably
so I'm amazed that nobody has done this yet, because this is the critical question. How many people of a certain type do you need to bring together to start an epidemic? That's the whole thing,
Well, I think the fact that we haven't seen it means it's difficult to get to work, right?
Yeah. Or maybe there's.
But we'd like to know infectious virus, and if someone's going to have to measure infectious virus,
plaque assays.
So, do we think that if they had continued this study beyond 28 days, the CT would get even higher as the immune response matured and developed, right? Or is this all we get?
I'm gonna place a bet here, and I'm gonna say no.
Your quarters
I'm
on the table, Rich.
I'm gonna say even after a boost, all right, that it's not going to make much difference, because I'm getting the distinct impression that even after one dose, right before the second dose, you're in pretty good shape.
Yeah, that's right.
That is what the Pfizer data said in terms of protection, was sort of day 12 seemed to be about where the difference came in.
So, does this match with the switch from IgM to IgG?
So what the other thing I was going to say is that if you look at Pfizer's phase 1/2 paper, where they actually show antibody levels, at day 21, which is the day of the second boost, and at day 28, the antibody levels are dramatically different.
Right.
And so I could imagine that I would sort of bet against Rich here and say that if they looked further, they might see a decrease because of that dramatic change in antibody levels that they're seeing.
What I would really like to see is data on this, and I'm sure their patient group will get big enough eventually after the second dose.
Yeah.
So
sure we'll see this.
Well, we want to see.
You get the reason they stopped at 28 days was presumably because that's when people were getting a second dose. And now the question is, if you follow from days 1 to 11 and 14 and 12 to 28 after the second dose, then what do you see?
So you need to do this for Johnson & Johnson's vaccine, then.
Yes,
a dose.
That too. That's the single dose.
So do we know what fraction of vaccinated people are getting infected?
Well, I ran the numbers on this I'm just assuming that they're looking at this whole 600,000 population, and they've got like 1,700, somewhere between 11 and 1,700 in this later group. That's 0.2% of that population. If you include all of the people, 0.2%.
All vaccinated people will get reinfected.
Okay. So that's what they're seeing. That's what they might be seeing in this sample. We would need the comparison number for the controls who are unvaccinated. What's the baseline population infection rate?
Because we don't know what fraction of vaccinated people are getting infected.
So
right?
We know how many they found here, but we don't know if that was.
So this is an ethical question. Would it be ethical to conduct an infectivity study among people who have already been vaccinated?
Yeah, they're going to
there is discussion about challenge trials going on now.
Yeah, I know that. You could take 1,000 people that have had the second dose of vaccine and just stick them in a room together and throw the virus in there and see how many people get infected. Because they're not going to get sick.
You could measure infectious virus shedding.
Yeah.
That might be. So, I don't like challenge studies with unvaccinated people, but.
No, me neither.
with two doses, which we know 96% efficacy at preventing COVID.
Yeah.
It might not be a bad thing. Although remember, there's always someone who doesn't respond. So
You'd have to test everybody first before you put them in the room.
Yes.
Then you need the monoclonals handy to make sure that
we do have those. But that would tell you.
The study could be done.
It's a good idea.
Thank you.
Would you volunteer for it, Dixon?
Sure, I would, because I need to know that answer, because I would like very much to go to dinner with some friends of mine who have also been vaccinated without having to wear my mask.
With no offense intended, Dixon, I think it would be best to do that trial in younger people.
Yes. No, there's
because they're right. Because they're even if the vaccine
right.
didn't take in them, they would be less likely to develop serious.
I would agree with that.
Because look, what we see here is that these individuals who had PCR tests done, they're clearly been infected after immunization, after vaccination, right?
Yeah, but it was a trivial.
But that was after one dose.
It's after one dose, I agree. So we need to know after 2 doses, but we also need to know what fraction of the vaccinated people are getting infected after vaccination.
And how infectious is that virus?
Did anybody who did get infected complain about being infected, or were they just randomly found out?
They don't really tell us.
You don't know, and that's an important question.
Right. So these are people who did for some reason get tested.
Right.
And I don't know what the standard is in Israel, whether they've got random testing going on or whether this is only symptomatic people.
That's another question I had, is how many of these were symptomatic?
I think Israel's testing game, I think, is pretty on point. So they may very well just be doing, this may just be like you showed up to work and that's why you got tested or something. I don't know.
Right.
Wow, this is quite interesting.
We've made some progress.
It is. It's a great start on this question.
Yeah, we'll have more, I'm sure.
Yeah.
We'll see what happens. All right. Let's do some email here before we do some picks. And let me take this first one. It's from Abby. She writes, the fan letters I've written in my head have...
I want you to start reading the letter, and you just go right ahead. I have something to do while you're reading.
Oh, yes, right.
This is a 2-person letter. Go ahead.
The fan letters I've written in my head have accumulated to the point of fug it. I just shoot off a note. So, Dixon's breathing is fine. It's part of the podcast. It's not about production quality. It's not about mixing or equipment. It's part of the podcast. I love it because it's part of what I've grown to know as the sound of comfort and new friends. And if the humanity and modesty were edited out, it wouldn't be TWiV. There are 45 to 48 weightier topics. I'd like to know where those numbers that I could send my first email about. But happy new weird. Vincent, Rich, Kathy, Brianne, Dixon. I forgot Alan.
Oh, well.
It's just an omission, I'm sure. Thank you for being my friend. Sincerest gratitude. Abby is a training coordinator for UT SWARP's Software Development and Systems Engineering Unit. UT, is that University of Texas? Could be.
Yeah, but SWARP's? Huh?
Anyway, there you go, Dixon. You're allowed to breathe. And you're muted.
Thank you so much.
Oh, that's University of Tennessee, Social Work Office of Research and Public Service.
Ah, good.
UT.
Okay.
Go Vols.
Rich, can you take the next one? Because it's from Texas.
Ray writes, Dear TWiV team, something occurred to me laying in bed last night, which appears to be a contradiction. I have learned from y'all that the vaccine is intended to prevent disease, not necessarily infection. Hence, Dr. Fauci has spoken about continuing to wear masks once vaccinated to prevent transmission. However, I've also heard Fauci mention reaching herd immunity through vaccination of a certain proportion of the population. What doesn't make sense to me is how so-called herd immunity can be achieved if vaccination does not necessarily prevent infection or transmission. What am I missing? Any help in correcting my thinking or explaining this logical inconsistency would be helpful. I am respectfully yours, Ray from Texas. So, Ray, you are justifiably confused. I think everybody's confused about this. And I don't know if I can even articulate it right. I do, however, in my mind, refer to our conversation with Paul Offit last week, where he had a more sort of nuanced perspective on herd immunity that wasn't so like absolute that, if a certain fraction is protected, then there's not going to be enough virus to infect all the rest of them. It's a little more subtle than that. Like my sense is it's like if you can, even if there's still virus circulating, if you kind of tamp down the load enough with enough immunity that I'm not doing well with this. that it's going to protect the uninfected, but that's the same. I'm saying the same thing as herd immunity.
I think Paul Offit's idea was that herd immunity is really about protection from seeing disease that affects the healthcare system.
Yes. Okay, good. Yes.
And so the idea is that, we may see some level of infection if we were doing super lots of PCR on everyone, but we aren't going to be seeing cases in the hospital. Ray mentions that he says the vaccine is intended to prevent disease, not necessarily infection. I would say that they checked to see whether the vaccine prevented disease. That was what they measured in the trials. And we don't know yet about prevention from infection. And that's what we're trying to figure out. So it could prevent from infection. It could reduce transmission. We just don't know yet. It wasn't like this was a conscious choice. It was just, that's what we've measured so far.
I would say there's a lot of hopefulness attached to this. So the idea when Tony Fauci talks about herd immunity as a concept, he's hoping that is possible. And he ballparked a number, and he's been very transparent about the fact that this is just basically a number he picked out of a back of the envelope type of calculation. They just don't rely on that 70% or whatever. And if the vaccine, Ray, you hit it right on the head. If the vaccine does not prevent people from getting infected, then no, we're not going to get true herd immunity in the absolute sense. And the 70% who are immune are going to spread it to the 30% who are not. And what Paul Offit was saying is if that infectable 30% is healthy enough and young enough and small enough a group, then we won't overwhelm the healthcare system. So we may very well still have people showing up at the hospital with COVID-19. We may probably have people still dying of COVID-19, but hopefully it won't be nearly so many, and we'll get through this by ultimately immunizing as many people as we can.
I would say that if 70% of the population has been infected, that I would guess that there's a significant fraction of them that are not going to get infected at all, or only very rarely because they're immune. And those that do will likely have a decreased viral load. Yeah. Those things combined make it so that the 30% who have not been infected are significantly less likely to encounter an infectious dose of virus. And so it's not black and white, it's gray. No. But it's effective.
And the paper we just talked about is trying to look at viral load as part of that question. So even if you're not completely protected from infection, if your viral load is low enough that you're only got a few hour window of infectivity and you're asleep, then you're not transmitting the virus. So effectively you're protected.
Yeah. So
I've accumulated 3 things that I want to say and haven't.
Yeah, go ahead, Kathy.
Go for it.
I agree with Brienne's analysis of what Paul Offit said regarding this being important from the standpoint of reducing serious disease that's going to be a problem for the healthcare system. And the second point, it was on February 4th that I most recently heard Tony Fauci's estimate, and it was 70 to 80% for herd immunity, and that we are somewhere less than 20% at now. And then something that's only kind of related to this, but this idea about, we need to continue to wear masks because we're still going to be able to transmit the virus even once we're vaccinated. And that while that's true, the emphasis on that starts to become problematic from the standpoint of people saying, well, then why should I even get a vaccine? If nothing's going to change. And so we have to present it in a way that says, yes, you yourself will be protected. And we think, although we don't know, that there may be less viral load and so forth that you will be spreading. And so there is value in getting this for yourself, and most likely there's value for the population. But if we want too hard on you're going to have to wear masks forever and we're never going to be able to travel again, until the whole world is vaccinated, we're going to reduce the number of people that are going to get the vaccine.
Good point.
Well, Kathy, there's an excellent article in today's New York Times that exactly addresses all of those issues. And we'll never achieve a 70% vaccination rate at the rate we're going, because a lot of people are refusing this vaccine, and that's unfortunate. And
However, some of them are really just hesitant, and they didn't hear Paul Offit's answer of how did we get this so fast? And it was money. That's important. And I know, or there's anecdotal evidence that among the healthcare workers who have been hanging back, now that they see other people getting it and doing okay, and as time goes on, they're maybe going to go ahead and get it. So that hesitancy may change.
And I think there's also an element of the hesitancy that's a little bit of sour grapes, because most people just can't get the vaccine yet. The demand is so high and the supply is so short that it doesn't really matter if you're hesitant.
Yeah.
Apparently, though, millions of doses are being stored in freezers and not used.
There are logistical and management issues.
So what Kathy just said that's the biggest reason, is that why should I get vaccinated when I don't even have to change my behavior no matter what?
Right.
Yeah.
So that's the biggest issue, I think.
I think,
but because it'll save your life is a fine reason.
I would add that. Yes, that's a good reason.
This is an unusual situation, because this is really the first big vaccine trial where we're doing PCR to see what's going on. And I don't remember another trial where we used PCR for, all the ancient vaccines, MMR, polio, et cetera. We just looked at disease and seroconversion. And I just remember what we're looking for is preventing disease. So the polio vaccine, we looked at paralysis. How many kids got paralyzed in the vaccine versus the control group? We didn't look at virus shedding. And it turns out that inactivated polio vaccine doesn't prevent infection and shedding. Yet, you get, I don't know what the number is for polio virus, but you get that. And we can achieve 90% of the kids in this country immunized with polio vaccine. That's enough to prevent everyone else. And one of the ideas about herd immunity is that the susceptibles who don't get vaccinated are scattered. They're not all in one place.
Yes.
Right? Because if they are, that's a problem. And the anti-vax people who don't get measles vaccine, they're all in one place. They tend to be like the religious communities up in Westchester, and they get outbreaks of measles.
Exactly.
No, and it's important to remind people that this post-vaccination masks thing is going to be a moving target. It's going to change. It's certainly temporary, okay, until we reach whatever sort of equilibrium we're going to reach, and then we just go with it, okay. But my guess is that the rules and the recommendations will change as we go forward and understand more about this.
Yeah, they will evolve.
Kathy, can you take the next one?
Kathy writes, "Love you folks so much and the important work you do. I always learn something even when most of it flies by 10 feet over my head. Can you tell me, without laughing at my possibly stupid question, what would happen to someone if they were to receive a first dose of, for example, the Moderna vaccine, but then follow it up 4 or 6 weeks later with the AstraZeneca vaccine? Might it nullify the immune response initiated by the first brand of vaccine? What would likely happen in this circumstance? I realize that due to lack of data, there wouldn't be a definite answer. I will be happy with a supposition or a probably. Thank you so much." So it's not a stupid question. It's one that many people have asked, even from the standpoint of what if you got one mRNA vaccine and then you got the other one? And we think that that's going to be okay because they're both basically almost the same identical messenger RNA molecule and so forth. Following up an RNA vaccine with the AstraZeneca vaccine, probably, that's the same antigen in both cases. It's the spike. So you would probably get a boost with that second dose. It shouldn't nullify the immune response initiated by the first vaccine in any case. So that's what I think.
Yeah, I've been trying to think of what could go wrong, right? If you did mRNA and adenovirus vectored vaccine, and I can't think of anything. And it's the right antigen, right? Can anyone think of something going wrong?
I think there are actually trials that have been done, not for SARS-CoV-2, but totally different in development of vaccine platforms where they have given people like an mRNA followed by an adenovirus or an adenovirus followed by an mRNA. Yeah. And the logic behind that is you may have pre-existing immunity to the adenovirus, so we don't want to re-inject you with the same adenovirus. So now we'll give you an mRNA vaccine. So it would not be breaking any new ground in that sense. And I would like to point out there are trials now being set up to look at some of these combinatorial possibilities. So in fact, I love it when listeners write in and say, oh, this is probably a stupid, obvious question. And then they ask the question that really needs to be asked. And this is one of those cases. Yeah, great question.
I've got it right here on the University of Oxford News and Events, dated 4th of February, 2021. They're recruiting for a trial with 8 groups. AstraZeneca twice, Pfizer twice, AstraZeneca plus Pfizer, Pfizer plus AstraZeneca for each 2 different intervals, no placebo group. Okay, so that's how good a question it is. There's a trial ongoing.
Yeah, and I think I heard that the Russians wanted to pair with AstraZeneca and do a 1-2 punch kind of thing. Yeah. So, 2 other things that I know that are going on are that Moderna has a trial of people 12 to 16 for sure. And I know that they're enrolling, one of the hospital systems in Detroit for a 2-dose of whatever is the 1-dose one.
Johnson & Johnson.
The J&J.
Yes, the J&J. Thank you. So they're enrolling to do that.
So I'm going to make another bet.
You've already lost a quarter.
Somebody ought to be keeping track.
And that is that down the road, years, when if in fact we're still vaccinating people against this disease, there may be variants that come along. Your immunity may wane or something like that. You'll get some sort of heads up from your doctor or your healthcare system and say, you really need to get a boost of your coronavirus vaccine, and they won't know what you got before, and they won't care what you get this time.
This happens with the flu vaccines. You get a different one every year, right?
Exactly.
And I think Brienne was going to say all the HIV trials, or many of them are different first and second doses, right, Brienne?
I have seen quite a few trials with the heterologous prime boost, yes.
Of course, now that's not a ringing endorsement because none of those have worked.
None of those have worked, yeah.
But yes, that's true. I mean, every year I look at what flu vaccine is different from the manufacturer and so forth. So
not just the composition, but the manufacturer, the platform, all that. Yeah. And they never ask the question. I always look. I want to know. I'm probably one of.
I always do too. Yeah.
Yeah.
But, I was excited this year when I found out after the fact that I had gotten the cell-based flu vaccine for the first time, but I didn't know until I got home and read the paper.
What is that? FluCellVax?
I think that was what it was.
Is that the name? Dixon, are you there? No, Dixon's gone. All right. Brienne, can you take the next one?
Sure. Kathy writes, hello, everyone. The shows get better and better. Re naming an episode, I suggest Viremia number 5 as a title to the tune of Mambo number 5. And Kathy is from Iowa.
We have 2 Kathys in a row.
I think it actually was Virema because we talked about some test or diagnostic or something the other day.
So I won't correct the spelling. Okay.
Yeah.
Oh, it's Virema?
Remember you thought you said something about how that should be a show title? You called it Virema.
No, I remember that. Yeah.
I didn't. I thought a show title of Viremia number 5 would be cool, but Virema. And Mambo number 5 is a very catchy song.
It is. I went through some of these emails before the episode today. And there have been multiple times where people have said something and I've wanted to quote the song Mambo No. 5, but I didn't think it would make sense unless we read this letter.
I went
first.
to look at the...
But it's been in my head ever since I...
So if viremia is what I think it is, it's capital VI, capital RE, capital MA, which is for viral recombination mapper.
Oh, that's right. That's right.
Yeah,
That's the
we
paper.
were talking about recombination.
Yeah, we did a paper on that. Exactly. That was the Dennison paper. Yeah. Exonuclease. Oh, yeah. That was a good title. Virema. We thought of just virema. We couldn't figure out what to pair it with. I think, Alan wasn't there, so.
I don't know if I would have come up with virema No. 5.
Actually, you came up with the title for that episode, Recombining Vaccine Doses or something like that. Oh, cool. Very good. I tried to change the lyrics of Mambo No. 5 because it's the way it is now. The guy is like hanging out with 5 different women. It's kind of weird, but I tried to make a different virus for each. On Monday is so-and-so, Tuesday. I stopped because it's not my talent. Dixon, you back?
I'm back.
Okay, can you take the next one?
I can. Rich writes, your show is fantastic. I listen every single week to both the podcast and the clinical update. There was discussion on a recent show that serology might not be positive if you've been vaccinated. Just wanted to give you a heads up that at least one serology test for spike diasorin liaison, PDF attached. That was probably a murdering of that word. I super enjoy everything I've learned from your podcast as well as the perspective. Your impact is huge. Best, Richard.
Diasorin.
And he's a professor.
He's a professor of everything, it looks like. Chemistry, chemical engineering, materials science.
So I want to tell you that I've joined a clinical trial for a post-vaccination where they're going to monitor blood for antibody levels. And when the person called me to enroll, I said, okay, I want to know, is this going to be a test for the S or is it going to be for N or is it going to be for both? And she said, I'm going to have to get back to you on that.
There is at least one.
Yeah. There are, as this person says, there's tests for spike and there are tests for N. And the one that is going to be our trial is just for the spike. They just want to follow our vaccine.
Right. Okay. There is one platform that I'm aware of. Roche has a test platform that's apparently built on test platform they've been using for years in clinical labs, that they now have tests for spike and for N on the same platform, which would be extremely useful at looking at this exact question. Right. Are you immune because you were vaccinated or are you immune because you were infected? Right. Just like we do with a Hep B test.
It's good. Very good. Alan, can you take the next one?
Sure. Dave writes, dear colleagues of the TWiV League. Okay. My understanding is that when one cell is infected simultaneously by 2 or more viruses and the progeny is a mix of the 2, this is called reassortment. I'm considering possible reassortment of Sniffles corona with COVID-19. Okay, I'll just read the whole letter and then we'll come back to it. Okay. Does this include any alterations in the genetic code or is it just a difference in the ensemble of glycoproteins that accessorize the capsid as the virus steps out? Are the characteristics of the reassortment predictable? When a large dog breeds with a small dog, one usually gets dogs of intermediate size. However, the parameters of a virus that are important would be things like tropism or recognition by antibodies or binding sites, etc. Since we would expect these to be very sensitive to nuances in the capsid proteins, a seemingly small difference in capsid glycoproteins could cause these parameters to change in unforeseeable ways. In your judgment, are Sniffles corona and COVID-19 similar enough that reassortment will lead to significant numbers of viable progeny, or would they be neither fish nor fowl and unlikely to result in further infection? Have you any historical examples of reassortments that you'd like to share? Thanks for all you do. Okay, so Dave, reassortment I think of as happening with viruses that have multi-segmented genomes like flu. And in that case, you get one virus coming in with 8 segments or 9 and another coming in with 8 or 9 segments, and you can reassort and come out with 8 or 9 segments in your new virus that gives you a different combination of segments. I think of a virus with a single monolithic genome, if you will, having a recombination, which is slightly different. It's not just a shuffling of segments, it's a movement of pieces of RNA into other pieces of RNA or pieces of DNA into other pieces of DNA. But that's really more of a terminology issue. I think the key question here is, could you predict and what might be the outcomes of these? And I think predicting it might be kind of hard, but it would depend on which pieces, which portions of the genome you recombined, right? I mean, if you recombine spike from another coronavirus, you might get a change in receptor tropism. If you recombined other portions, you might get a change in pathogenesis. And I don't know of, I'm going to have to turf this to the group, examples of coronaviruses doing a lot of this.
Recombination, I now, since you've corrected this to recombination, so I assume this last sentence would be, have you any historical examples of recombination that you would like to share now? Well, I mean, the SARS-CoV-2 is thought to be a recombinant. We've done a couple of studies which look at the lineage and say, yeah, recombination has gone on. It probably happens among all these coronaviruses. They exchange genes, spike genes, and others as well. Yeah, for sure. Many others, I'm sure.
And going back to his original question, then, could there be a reassortment of a common cold coronavirus with SARS-CoV-2?
Oh, did he ask that?
He did.
He did. Yeah,
Sniffles corona.
That's what he meant by sniffles.
Yeah.
I didn't know what he meant by sniffles corona.
Yeah.
That's
and
cute.
I'm still parsing that question. I think of all kinds of things. I don't know to what extent. I would assume at least some recombination amongst coronaviruses is dependent upon homology among the genomes.
Yeah.
Is it homologous recombination the same way we think of homologous recombination in DNA viruses? I don't know. Yeah,
I thought it was like a strand exchange. Yeah,
I kind of think of it as a template exchange with the.
But
there's that purple sequence, depending on if it's the textbook or the diagram that Vincent showed.
A lot of the recombination. I'm sorry. Sorry, Kathy. Go ahead.
I was just going to say, it seems like there's a sequence that is conducive, and then it is at the other end when it jumps. So if that sequence was similar enough in a heterologous virus, sniffles versus SARS-CoV-2.
So it is. I believe it is driven by some homology. They call it micro homology. Yes. In the paper we did about exo and recombination, they were talking about regions. Their favored regions of recombination, right? And the mRNA formation is basically recombination because the polymerase goes from one end of the genome to the other. And that involves micro-homologous regions. So the question is whether, I think it's limited to the same genus, but I might be wrong, like the alpha and the beta coronaviruses may not recombine across each other. And some of the sniffles coronas are one or the other, right?
Right. So when RNA viruses recombine, they don't do it like wildly higgledy-piggledy so that any polymerase goes from anywhere on one genome to anywhere on the other genome. Okay, I think there is this micro-homology that probably has in the picture that's in my head, there's a little bit of base pairing, okay, that is between the molecule that's being copied and the one it's jumping to that directs that exchange. But how much that has to be and how much the variation in genome structure amongst different viruses would affect that, I don't know. And somebody.
Well, I think there are actually two issues. So DNA viruses clearly it's homologous and you need a stretch of homology and you get a crossover event in holiday junction, yada, yada, yada.
Right.
But RNA viruses, there's this microhomology thing going on that you need some degree. And as Rich says, I'm sure somebody's doing a lot of work on exactly what that degree is and what controls it. But the other thing that occurs to me is you would also need, in addition to the microhomologies, you'd need compatibility of parts, if you will. So the limit from an alpha to a beta coronavirus might be partly about sequence and partly about just, that nucleocapsid won't work with this virus because it won't fit, right? Like you can't take parts from your Volkswagen and put them onto your Ford.
Yeah. And so to sort of put that into some of the language that Dave uses, perhaps you could make that alteration in the genetic code, but then when you tried to actually build the glycoproteins, they wouldn't fit together and the virus would not actually be able to persist and be selected. So it would be sort of selected against. I wanted to use the word viable there, and then I realized it was messy. So I suddenly had to switch.
Not quite alive, are we?
Yeah. So let me... Go ahead, Kat.
Go ahead. No, you're going to show something because maybe it relates to the purple sequence and the...
Yeah, I brought up the manuscript that we talked about a couple of weeks ago, the PLOS pathogens from Mark Dennison. The proofreading exo mediates viral recombination. And here they only look at beta coronaviruses, right? So I have a feeling that you can't recombine across beta and alpha. But they make this statement in the discussion. Now I lost it. Sorry, because I went back. Let's go to the discussion again. Sorry. Where does it say something about microhomology is involved? Recombination. I lost it. Well, you can see in this recombination map, I mean, these colored lines are the hotspots for recombination. So there are certain regions of the genomes that recombine and others do not as much. And I think that's driven by, see here, sequences containing microhomology are likely determinants of recombination resulting in defective viral genome, which is a form of recombination, right? So I think that's a big part of it. Whether there's any contribution of non-homologous regions, I don't know.
So I'm thinking that if there was really rampant recombination amongst the 4 sniffles viruses, coronaviruses that are out there, that it would be hard to maintain each one as a distinct entity.
True.
All right.
Yeah. Yep. I agree.
So this is a long way of saying we don't know.
We don't know. The answers to your questions, Dave, are yes and no.
And I know notice that we're going to have Susan Weiss on sometime in the future.
Great. Yes.
And if we remember, we can try and save this question for her.
Yep. Yeah. That's a good idea. I wanted back to answer fundamental questions about the reproduction of coronaviruses.
And there's another issue in this, and that is that even if you could recombine these viruses, the genes from the different viruses are not necessarily going to cooperate with each other in a fashion that's going to give you a productive infection. It's about more than just doing the not every recombination is going to be okay.
That's what I was getting at with the interchangeable.
A lot of them are just going to fail. Yes, right.
Now in pox viruses, Rich, is it homology driven?
Yes.
Always, yeah.
Okay. And it's you need about somewhere between 1 and 300 base pairs. Mm-hmm. And God, these viruses recombine like crazy. If you take 2 highly homologous pox viruses and do a mixed infection, you get a crossover. I think at least each kilobase. All right, they just scramble. The genomes just scramble, and yet you can have pox viruses that are sufficiently distant, so that you can do mixed infections with them. They're closely enough related so that biochemically they can help each other out. Okay, like you can prime an infection with a Shope fibroma virus, and then throw on vaccinia virus DNA, and the transcription machinery from the SHOP will activate the vaccinia DNA genome so that they'll help each other out, but they won't recombine at all.
Yeah, this recently in my course, I like to show the experiment where they reconstructed horsepox virus from 10 synthetic DNA fragments overlapping, and all they did was put them all together in the same cell, and they recombined, and boom.
Yep.
Out came virus.
They just zip together.
Amazing. Let's do some picks. Dixon,
Okay.
What do you have for us?
Another shout out to Rich, because I actually emailed him about 3 days ago to ask if he was okay, because the news was really disheartening to show how many people had broken water pipes, and they had a picture of a ceiling fan with icicles hanging off of it. And Rich informed me that everything was perfect. He was ensconced in his home. He had heat, he had water, and he had food. That's all he needed. Well, Rich, I would say that this is one of many events that will probably be happening over the next 20 years. Polar vortex is an unusual occurrence, but it now becomes explainable. And again, there was a nice article in the New York Times today about that. This is from the Weather Channel. It's most unusual for weather this cold to get that far south. Now, I'm wondering whether there were any winter crops that Texas is noted for, like grapefruit or something like that, that just got completely trashed.
Oh, yeah, gotta be.
Because and look at all the wildlife. Did you realize that there wasn't a single zoo south of the Mason-Dixon line that didn't lose animals to this event?
Oh.
So when you're thinking about, the cruelty of nature against humanity, think of what we've done to those poor animals by making them prisoners in some attractive place to visit, and then all the chimpanzees die, or all the giraffes die, or they're just not equipped to handle this kind of weather. And this will become a new normal, I'm afraid. That's what
I
I'm
hear
afraid
that
of.
the snakes were able to catch a flight to Cancun, though.
One of them, and its family. That's what I'm hoping happens to the Burmese pythons in Florida. But Florida didn't get this kind of weather.
Right.
They did not get this kind of weather. So that occasionally they do, but not this time.
Dixon, do you think in times of such natural disasters that the government should play a helping hand?
Well, I would think so, but to have a governor of a state blame it on renewable failures like solar panels and wind turbines, when Texas gets, 2% of its energy from those two sources, that's a charade.
I just want to point out that we've got solar panels and wind turbines all over New England, and we don't have these problems.
This is true, you don't. In fact,
y'all
you
down
have just
in
about
Texas
the opposite.
Just need to build them right.
Yeah, if you build them, they will survive.
Brianne, what do you have for us?
So I have a story that I came across yesterday that just was really cool. So there is an animal called the black-footed ferret that is an endangered species. They are down to about 400 or 500 members on a particular ranch in Montana, where many of them are. And one of them was just cloned from a cryopreserved sample that was in San Diego of a black-footed ferret that died in 1980, in the early 80s. There has been this worry that, the small number of these ferrets was getting quite inbred. There was a lack of genetic diversity and that the species would suffer from some difficulties. And so they are hoping to be able to take some of these cryopreserved cells and clone new ferrets like the one they have made so far. Her name is Elizabeth Ann, and introduce more genetic diversity into the population. And I just hadn't seen something that had been cryopreserved for quite that long be cloned, and it seemed just like a cool thing and also interesting for conservation purposes.
Just don't name it Dolly.
You think that'll give it bad luck, Dixon?
Well, it did for Dolly, that's for sure, because the cells that they cloned her from were old cells, and she died on premature death.
What did they do here?
Yeah, but she lived a life of fame.
She did. She did.
Her candle burned bright.
This is all true.
So too bad they didn't do this with the dodos, right?
Yeah, or the giant ark, or where you have a whole bunch of animals that we wished.
Yeah, but they do quote in here Ollie Ryder from the San Diego Zoo, And even from the time when I lived there, they were saving samples. And remember when I told you I saw the behind the scenes aspects at the wild animal park, I saw some things that I, imagined were perhaps part of those big liquid nitrogen tanks of biological specimens that they've been saving for years. So yeah, pretty cool.
Yeah.
Alan, what do you have for us?
I have an XKCD cartoon. I've picked a few of them over the years. This one is really good and timely. It explains how mRNA vaccines work in Star Wars terms.
It's awesome.
It's just a really good walkthrough of what the mRNA is doing, and represents how this plays out. The cell gets the blueprints and builds a Death Star because that was what it was told to do. And then the body, of course, attacks and just figures out how to destroy the Death Star. And then when the real one shows up, it destroys it.
It's funny.
Cool. Yeah.
The first time I saw this was right when I was teaching some students about vaccines, and I sort of looked at it and I was like, "Why did I even bother to make a lecture?"
Yeah. And with an XKCD, you should always hover the mouse over it because you get an additional punchline. And this one is: to ensure lasting immunity, doctors recommend destroying a second Death Star sometime after the first.
Great.
Right.
I love this panel. These blueprints are from Princess Leia. She's always giving us projects. I think that's great. I love it. Really cool. Rich, what do you have?
I believe I have picked this way out there in the past, but I'm repicking it partly as a public service announcement. This is a really pretty boring pick. There you go. It's a nested pick too. I talk about the Brighton collaboration all the time. This is an organization that was created about 20 years ago for the purpose of formulating international guidelines for vaccine safety. And so I've given a link to the Brighton collaboration so you can see what they are now. They're currently, they've gone through several different metamorphoses, and they're currently funded by CEPI, and they're sort of the, if you like, the safety consultants or safety arm of CEPI. I've also linked there to the committee that I work on that's now called Bravato. It's a benefit risk analysis of various vaccine templates. And what that committee does is to try and formulate, it has formulated, templates for developers to fill out that describe their vaccine platform, or it can even be used to describe the particulars of a vaccine. And we've now developed templates for all 5 platforms: protein, inactivated virus, attenuated virus, vector virus, which is the one I worked mostly on, and that's how we started, and mRNA template. And just to give you an example, and this, the reason I thought of this is because I spewed out all of this stuff about the AD26 vector the last time we were together, and the reason is because of this publication that I helped put together on the adeno 26 vector. Okay. And I linked it there as adenovirus 26 vector template. So this is what happens when you fill out one of our templates. You get this, and it's like a review in a way, and it tells you everything that you could possibly ever have wanted to know about the AD26 vector. Now, of course, the problem, especially in the middle of a pandemic, is getting other developers to fill these things out. They have other things to do. But the intent is clear. And in fact, this is a good example of how useful it can be. Because if you to try and understand everything that went into this template and how it works, and what sort of trials have been done and what the safety data are, you got to go through hundreds of publications, and it's all here in an organized fashion. And if everybody filled this out, it would be the same organization for everyone if you were familiar with the template. So That's just, if you're interested in Ad 26, there it is. If you're interested in the Brighton collaboration, there it is. And if there's any developers listening, think about it. Fill out one of these templates.
I have just 2 quick comments here. First, the Brighton collaboration logo is awesome. It's an antibody that's also a stick figure of a person.
Yeah.
Yes.
I think that's really cool. And the second thing is this template approach is a great idea. And it's very much like something that's used in aviation called a flight risk assessment tool, where people do all these studies on crashes and all its data goes into it. And then they come up with a form that the pilots go through. They say, okay, our wind speed is this, our runway length is this, and they fill it out and they come up with a number that says, this is your risk for this flight. So this is kind of what you're building here. And that's a great tool.
The vector template was particularly interesting. As I said, that's where we started, because in my mind, in order to understand how a given vectored vaccine works and its potential for causing problems, you have to understand the virus that it came from initially, and then what modifications were made to that before you even stuck a gene in it. And then when you stick a gene in it, does that change its properties any further? And how is the gene expressed? And what do you know about the safety consequences? A bunch of other questions as well. So all that's there.
Good stuff.
Very cool.
Kathy, what do you have for us?
I have something that's a little bit different. It's a short 4-minute read. It's by Dr. Connie Pechman, who's a marketing professor in the School of Business at UC Irvine. And the gist of it is that the title of it is how Facebook can make up for disinformation and help get everyone vaccinated for COVID-19. And the gist of it is that if you could use ads targeted to zip codes where vaccine uptake is low, it could potentially have positive results, especially if those ads would appear multiple times for the people in those zip codes. And so she has an example that I found compelling. She said, and I didn't click through to the study itself, but she says, I recruited participants for a quit smoking support group using Facebook ads that targeted zip codes with predominantly Black populations. Compared to a similar campaign that showed ads to smokers across the country, the zip code targeted campaign more than doubled the number of Black applicants for the group. And they did the same thing for Latino populations, and the number of Latino applicants more than tripled. So her thing is, if you could, get Facebook to help out and target public health ads to particular zip codes, it could go a long way toward getting better vaccine uptake.
That's a great idea. I really like that. Let's see if Facebook does the right thing.
Facebook could finally use their powers for good.
Yeah.
That would be awesome.
Yeah. Or if somebody had enough money to figure out how to get that same kind of database.
Yeah.
I don't know. Yeah.
It really ought to be a public service.
I bet you Philip Morris cigarette company knows already.
Well, of course.
My pick is an op-ed in the Times today by David Quammen. It's entitled, "And Then the Gorillas Started Coughing. Humans are spreading coronavirus to other animals. What does that mean for all of us?" It's quite nice. It's about how this initial infection of gorillas in San Diego, he says that for the Evening News, this was a cute animal story with a happy ending. For certain biologists and veterinarians, it was a small seismic tremor that reminded them of some ominous, little recognized possibilities related to our pandemic event, which has already been tectonic. And he goes and talks about, there are infections of various animals going on, which we should not ignore. And it's not just a matter of, so one of the suggestions has been great ape tourism be suspended and field research reduced until the health risks to these animals can be balanced against loss of income to local communities. And then he spoke with someone else. He said it's very easy to say stop going to the great apes until we've solved the problem. But if you do that, the apes are dead because they'll be shot. You have to have someone who's looking out for them. So it's not a simple solution. And he talks about a few other viruses that have been known to cause human viruses, including human metapneumovirus, which I didn't know has infected gorillas. So good writing and good point, I think.
Vincent, is there a term for this spillover from animals to humans and zoonotic? What would you say in the opposite of that?
Spillback.
It could be spillback. He calls it anthroponosis.
Yeah. Anthroponosis.
I have also heard reverse zoonosis.
Yeah,
so
I heard that too.
Reverse.
They all work.
And this came up very early in the pandemic on TWiV when we were discussing spillback into cats and dogs and other animals and bats from humans, and how this could this could actually make the virus endemic just because it'll constantly be spilling over and back.
Yeah.
And rodents and minks.
And rodents and minks, yes.
So, Alan, for gorillas, it would be spill silverback?
Possibly, yes.
Thank you.
So there is one example that he didn't cover, and that is a number of years ago, a group of chimpanzees in Uganda were found to be infected with rhinovirus C, type C, and it was lethal in some of them. And this is interesting because it was humans that spread the virus to them. And in humans, there's a polymorphism in the rhinovirus C receptor gene, which if you have the right amino acid change, you are more resistant to serious disease. And none of the chimps have that resistance polymorphism. They're completely susceptible. They get serious disease. Why? Because the virus is not selected for it. It doesn't circulate in chimpanzees. It's a wonderful example of how if a virus circulates for a long time in humans, you may select for changes that mitigate disease. And the chimps never seen it, doesn't circulate among, but the people bring it to them, and it's highly lethal. So I sent it to him, and he really liked that. That was cool. All right, we have a listener pick from Kim. It's a photo of an ice sculpture called "Virus Getting the Shot." And I think it's just gorgeous. It's an ice sculpture of a coronavirus characterized with a syringe going in its head, and it's very unhappy.
Yes.
It's got...
it's not a syringe. It looks like a sword from here.
I thought so, but it's got lines on it,
Yeah, no, you're right. You're right.
Like a syringe would have. Now, you see the spikes, they're there, but then there are these other things shooting up. What do you think that is?
I think it's blowing up.
I think it's
I
lysing.
think it's the guts coming out. Yeah.
Oh, okay. It's gorgeous. It's really well done. I think we'll make that the thumbnail for this episode. All right, that is TWiV 722. Oh, cool. This is from Kim Without Borders. She puts on the bottom of Kim Without Borders, Dentist to the World. That's so cool.
That's good.
I like that. Dixon, that's like Parasites Without Borders, right?
I hear it. Absolutely.
Okay.
And Bats Without Borders.
TWiV 7. You're going to get email about the bat story, Dixon.
There's no bats about it.
722. The show notes are at microbe.tv/TWiV. If you want to send us a comment or a question, TWiV at microbe.tv. And if you'd love to support us, microbe.tv/contribute. Dixon Despommier is at trichinella.org, thelivingriver.com. Thank you, Dixon.
Another enjoyable day. This was very educational and collegial. I loved it.
Kathy Spindler's at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Brianne Barker's over at Drew University, BioProf Barker on Twitter. Thank you, Brianne.
Thanks. It was great to be here.
Rich Condit is an emeritus professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
And Alan Dove is at alandove.com on Twitter. Alan Dove. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.blog. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.